CO2FOREARM is a European-funded project aiming at providing fundamental understanding and simulation technology for CO2 storage through microbially enhanced carbon mineralization, a technology that has the potential for switching the paradigm of geological carbon storage for climate change contrast. The project focuses on carbon mineral storage, which is a viable alternative to conventional geological carbon storage. Carbon mineral storage consists in injecting CO2 in highly reactive mineral formation where it can react with the resident rock and therefore be stored permanently. This has two main benefits: availability and security. Mineral formations are found globally, and it has been found that CO2 can fully mineralize in 2 years in such formations. The particular interest of CO2FOREARM is in microbially enhanced carbon mineralization. Biological catalysts are used to alter reaction rates and further enhance carbon mineralization. Biological catalysts enhance carbon mineralization mainly by two mechanisms. Firstly, they contribute to increase the pH of the CO2-rich aqueous solution, hence creating the favorable conditions for carbon precipitation. This process is called bioalkalinization. Secondly, they act as nucleation sites by retaining the calcium ions in their surroundings, therefore creating a microenvironment where carbon precipitation is more likely to occur. The processes involved are extremely challenging to model due to their fully coupled nature. Indeed, fluid flow is altered because of the biofilm dynamics. To the same extent, the biofilm growth morphology is also dictated by the flow conditions. Reactive transport also plays a dominant role by providing the necessary nutrients for biofilm growth, and at the same time, biofilm induces chemical reactions such as the bacterial ureolysis and the bioalkalinization. Not only the processes are fully coupled, but also they interact with the porous microstructure, inducing changes in the porous microstructure such as bioclogging and dissolution precipitation. The major challenge of CO2FOREARM is therefore to model, both at the conceptual and the numerical level, these complex process-structure interactions. The specific objectives of CO2FOREARM are to develop or scale coupled biogeochemical reactive flow simulation models, to assess the use of biological catalysts to enhance carbon mineralization, and to produce correlations, relationships for use in larger scale simulations under uncertainty. The core of the research lies in the development of the simulation models. For that, we have developed and released in GitHub an open source software named BioReactPy, which is specifically developed for simulation of microbially mediated multiphysics processes in the subsurface. These tools allows us to simulate the complex biofilm, fluid flow, reactive transport processes happening in the subsurface at the scale of a single pore. You can see here an example of such a fundamental study investigating the coupling between fluid flow and biofilm dynamics for different hydrodynamic conditions. The second step was to apply these models to assess the role of biological catalysts in enhancing carbon mineralization. For that, we set up a simulation test case relevant to carbon mineral storage and performed simulations with and without the biological catalyst. Our results clearly indicate the bioalkalinization effect, that is, the enzyme urease present in the biological catalyst stimulate the bacterial ureolysis, which in turn increases the pH of the aqueous solution. This creates the favorable conditions for carbon mineralization and leads in a highly nonlinear way to enhanced calcite precipitation. The third step was the validation of the numerical models with experimental data. For that, we relied on a unique dataset documenting absolute reaction rates of a calcite sample. Experiments of calcite dissolution were performed at Politecnico Milano using atomic force microscopy. We developed a numerical approach that is consistent with the experimental workflow and documented convergence for vertical resolution down to sub-nanometer resolution. The main scientific outcomes of CO2FOREARMs are the development of original simulation models tailored to the analysis of pore-scale microbial mediated reactive processes, and the release of a thoroughly validated open source software on GitHub. With the aid of these models, CO2FOREARM documented the highly nonlinear dependency between urease activity within the biofilm, pH and amount of mineralized calcite, We documented the existence of a critical hydrodynamic point where biofilm transitions from bulky morphology to a boundary layer diffusive one, and the existence of different bioclogging accumulation patterns suggesting that different representative elementary volumes may exist for effective porosity and permeability. Finally, we validated for the first time a continuum scale model against high-resolution experimental data of calcite dissolution, which is an important milestone in geosciences and physics of fluids. CO2FOREARM had substantial impact on the scientific community in several aspects. It advanced the understanding of biogeochemical reactive processes in the subsurface. It provided quantitative description of the impact of biochemical processes on carbon mineralization. It advanced the open source simulation technology for pore-scale microbial mediated reactive processes in porous media, and specifically the open science policies and results reproducibility implemented were crucial to maximize the scientific impact of the project. This allows other researchers to exploit the multiphysics models to answer other engineering questions arising in the subsurface, for example, for code comparison or benchmark validation.